So the next presentation is by Altan Kokatulum. So Altan, wherever you are ready.
All right, can you all see and hear everything okay?
Yes.
Okay. All right, hello, my name is Altan Kujatalam. I'm from Alfred University, and this is my REU project on improving Alpaca, which is a program standing for Automated Landmarking through Point Cloud Alignment and Correspondence Analysis, which will make a bit more sense soon. My mentor for this project was Dr. Porto from the biology department at LSU. So this program has to deal with the field of geometric morphometrics, which is analytic techniques that biologists use concerning specific anatomical points of interest on a structure. As you can see here, we have two different skulls, which have all these little points on them. So they will take the coordinates of those points and do various mathematical analyses with them to test hypotheses about how these structures evolved and how they function within the ecosystem. So how the program essentially works, it is to automatically put those points on the structures. Usually people would do that manually, which can take a while. And so this is a way to make the data acquisition part of these studies much easier and faster. So how Alpaca works is we start with two samples, one of which has been annotated manually, has all the landmarks on it, and then we have a second one which we want to get the landmarks for. So we have our two samples here, and then we are going to downsample them to get point clouds instead of the full 3D mesh. So then we're working with much less data, which allows the program to run faster, and it doesn't seem to have a significant adverse effect on the accuracy. Then it does a rigid alignment, which is essentially rotating and translating the structures until they are lined up as best as possible. And then the deformable or non-rigid alignment here, it will sort of squeeze and stretch the initial source model, which has the landmarks, to match the model that we're trying to get landmarks for, so that the shape is matching the second one, but all the landmarks are preserved. And then it will just transfer the landmark points onto the new model. And so that non-rigid point cloud registration step takes the longest. It is a very significant mathematical undertaking. So that is the main point we are trying to improve here. It uses a coherent point drift algorithm, which imposes constraints on the points in the cloud so that they can only move in specific ways, and that one point can't move too differently from the points around it. And it's based on some obscure mathematical concept called Gaussian mixture models. We don't have time to get too into that. So the change that we made, there was a new coherent point drift algorithm that was published just last year, and it runs in a Bayesian setting. Essentially means that the underlying assumptions are looser, which allows us to take some shortcuts in a very broad sense. So we can accelerate this with various methods in linear algebra with the Nystrom method and KD tree search. And here we can see that we get to similar levels of error in about the same steps, but it is much faster. The BCPD is the new one, so the orange lines have a much faster computing time than the old method, which are the blue here, and also scale better with the size of the point clouds. Like this one is with 100,000 points, whereas this one is the new method with 100,000 points. So there's a much bigger increase from 50,000 to 100,000 here than between these two. These are what the algorithms look like, but we definitely don't have time to explain those. So this is what the program looks like. We have our 3D model here. You can see these little pink dots on there are the landmarks. You can rotate it around, look at it however you want. Over here is where you tell it what you want it to do. So we can pick out the source file here, the landmarks for the source file, and then a folder for all the targets that we want to find landmarks for. and then the folder we want to put our outputs in. There's a few advanced settings you can fiddle with, but it's pretty straightforward. You just pick out your files and then you click the run button, and you can see all the magic happen over here. And this is within 3D Slicer, which is an open source platform used in all sorts of biological modeling settings. And it's free, relatively lightweight. It's handy for a lot of things. And then since we were able to get it, use this faster algorithm, we are then able to run the comparison between multiple source models for each target. So instead of just running one pair, we run the same target with 5 different sources. And that allows us to get more accurate results because of the variation between the source models and the targets and all that. And so essentially we just run the whole pipeline for the 5 different sources, and then we take the median of the results. And we can see here that that improves the accuracy. This is a graph of the error versus the number of sources. So here at 5 sources, we can see that the error is quite a bit lower than for 1 and 3. The variance is also somewhat lower, and that going to more than 5 sources doesn't seem to help much. So 5 is the sweet spot there. Here's a more detailed comparison of accuracy. We see the median error over here for the original version is a little bit higher than the median error over here for our new version with the new algorithm and using 5 sources. So this is for each landmark in our data set. Each sample has 45 different landmarks in it. So each box here is the error for each landmark. Then the different colored points here are to compare with other methods. So blue is another automated method that has been published. We're generally under there, which is good for us. And then green is the error that you would expect for different people putting the landmarks on manually. So we're taking that as the minimum we could possibly hope to achieve. And we're doing pretty well in that capacity. Also worth noting that, again, the variance is lower as well. It's not just the median error with the new version. Here is more showing how we can go faster with larger point clouds using the new algorithm. As you see, the slope of those is much less than for the original here. So sometimes it's possible that using a larger point cloud would be helpful. In the data set we've been using, 5,000 is sufficient, but if you have a more complicated model, we might need more, and it's nice to have that option. And yeah, that's what I've got. Any questions?